Welcome back to the lecture on autonomic nervous system. We have covered quite a few topics so far: the anatomy of the sympathetic and the parasympathetic divisions, the biochemistry, that is the receptors and the neurotransmitters that are involved. Now, we also talked about the two different levels of activation of these two divisions, the tonic and the activated levels. Now, we would look at the two different types of reflexes that are possible. That is simply because you have a preganglionic neuron that originates in the CNS, postganglionic neuron that originates in the peripheral nervous system, and so that creates a shorter loop. The preganglionic neuron being involved creates a longer loop. So if you receive a sensory signal in a long reflex, it would be carried to the CNS, that is through the spinal cord, maybe the brain structure, and then the processing of this information. And the preganglionic neuron, it's a motor neuron that originates in the CNS and then synapses with the postganglionic neuron and then ultimately affect the target structure. So the signal is carried to the CNS, the response is communicated via two different neurons to the effector. So that's a long reflex. And the short reflex could be the stimulus that is being taken to the postganglionic neuron, bypassing the CNS here. So the postganglionic neuron can respond to the stimulus, typically when it has to be a quick response. This type of response, both the long and short reflexes, are commonly seen in the digestive system. Some functions like the local churning, motility, secretion of hormone, those can be through the short reflex, and the stretch and the secretion of a variety of substances related to digestive function may depend on the long reflex. So both are utilized, but this is most commonly seen in the digestive system. All other systems that we associate with the sympathetic and parasympathetic divisions typically use a long reflex. Here comes a very detailed summary of the types of receptors in the autonomic nervous system. The neurotransmitters, they typically bind to, and the structures where you would expect these receptors to be present, the nature by which they respond to the stimulus, like the neurotransmitter binding, and what type of activation do you see at the target cell? Is it ionotropic or metabotropic? Is the kind of division we have here. And is it an excitatory effect or an inhibitory effect is the last detail here. So, for example, let's talk about the nicotinic receptor. Nicotinic receptor is divided into N1 and N2. N1 is not part of the autonomic nervous system, but I just wanted to show it here. Skeletal muscle fiber has N1. So this is not part of the autonomic nervous system. Okay, it's just to compare and show you that there is another type of nicotinic receptor. Now, all of the postganglionic neurons would have N2. All autonomic postganglionic cells and adrenal medulla both have N2. So that is the most common type of receptor in the autonomic nervous system. Okay, and this is through ionotropic activation, that is the opening of an ion channel, let's say more sodium ions than the entering the cell than the potassium ions leaving, therefore depolarization and therefore the excitatory effect at the postsynaptic cell. So that is a function of nicotinic receptor. Now, muscarinic receptor is seen at the target tissue. It is also divided into many subtypes, M1, M2, M3, etc. M1, for example, is on the sweat gland. M2 is specifically in the cardiac muscle. M3 is again on some exocrine and some endocrine glands. So those are all target structures. That's where you expect one or another type of muscarinic receptor. So acetylcholine binds to muscarinic receptor at the target cell using one of these receptors and use metabotropic approach, that is G protein activation and then the subsequent activation of IP3 or calcium channel opening or the cyclic AMP mechanism. And the effects can be excitatory or inhibitory. In a simple example, like the heart, you would expect the binding of acetylcholine to M2, bringing about decreased heart rate, decreased contraction force. Whereas at the other target tissues, this could be the decrease in the secretion of a particular juice or increased secretion of sweat. So those are some effects that you would expect to see with the activation of muscarinic receptor. Alpha 1, alpha 2, beta 1, all three bind to both norepinephrine and epinephrine. Beta 2 is more specific to epinephrine only. All right, so where do you find alpha 1 receptor is where you need to know that it is in the lining of the blood vessel, that is the smooth muscle lining of the blood vessel. And then many other structures. These are tissues, not blood vessels. You would have alpha 1, and it again uses metabotropic mechanism and brings about excitatory effect. So that is the increase in the contraction of the smooth muscle causing vasoconstriction. So that is considered excitatory. All right. Now, alpha 2, as I mentioned, binds to both norepinephrine and epinephrine. It's simply known as an inhibitory type of receptor. Whenever you find it, you may see the presence of alpha 2 in certain parts of the digestive structures, in the smooth muscle lining again. So this can be confusing because you have smooth muscle here and smooth muscle here. So yeah, certain parts of the autonomic target tissues would have alpha 1 and certain structures would have alpha 2. Adipocytes, platelets are distinct ones, and that's where you would cause vasodilation instead of vasoconstriction. So that is because of the relaxation of the smooth muscle, not the constriction effect that you would see here. So that is via inhibition of the cyclic AMP pathway, unlike in some instances where you have the IP3 DAG, and that is the activation of the muscle contraction. Correct? So now here, this is the relaxation of the muscle. Beta 1 is specifically in the cardiac muscle, and its mechanism of action is through the activation of cyclic AMP and the increase in the muscle contraction. That's how it could trigger increased force of contraction, increased stroke volume. It also acts at the SA node and AV node to bring about increase in the heart rate. Beta 2 is specifically in certain areas like the bronchioles, lining of the airway, and then the smooth muscles of certain arterioles. This causes bronchodilation. Okay, a bronchodilation by causing relaxation of the smooth muscle. Similarly, relaxation of the smooth muscle of certain arterioles to bring about the opposite effect of vasoconstriction, that is vasodilation. So vasodilation or bronchodilation could be the functions you would assign to beta 2. All right, with this information, now we will look at the organ systems that the autonomic nervous system would affect, and the summary of what some of the major effects of the activation of autonomic nervous system would be. So here is a list of the autonomic structures. And now we compare the sympathetic action and the parasympathetic action. We also bring in the receptors that could be associated with the sympathetic or the parasympathetic systems. All right. So the parasympathetic system is via the muscarinic receptor at the target structures, whereas the sympathetic would be through alpha or beta receptors. There is one exception, that is sweat gland, that is supplied by the muscarinic receptor. So this is a rare example of sympathetic system using muscarinic receptor, but that is still true. All right, so that is one exception, whereas elsewhere you would see the alpha or the beta receptors being involved with the sympathetic system. All right, so looking at the heart, it's very straightforward. A sympathetic activation increases heart rate, increases contractility. Increased parasympathetic action would bring about the opposite effects. And we already discussed the receptors beta 1 in the heart for sympathetic response, muscarinic receptor, specifically M2 for parasympathetic action. This is the next one can be confusing. Vascular smooth muscle. So certain areas would have alpha 1 supply, certain areas would have alpha 2. So that is where it can be confusing. So let's take a look at constriction of blood vessels in the skin. All right. So now during low temperatures, in order to conserve heat, we may want to constrict blood vessels in the skin, and that's where alpha 1 receptors would be activated, and dilation of the blood vessel in skeletal muscle. All right, so this is very confusing for students. Sympathetic activation, there is increased blood supply to the skeletal muscle. All right, please note that skeletal muscle is not part of the autonomic nervous system, but we talk about it because it's one of those physiological responses that you see. And how do you explain that? Why is there increased blood supply to the skeletal muscle? That is because skeletal muscle has beta 2 receptors. Beta 2 receptor activation causes vasodilation. That is how you would increase blood supply to the skeletal muscle. So important detail, although this is not this is not an example of an autonomic structure. Now, gastrointestinal tract, you have all kinds of receptors, and then you have sphincters, you have smooth muscle lining the stomach, the intestine, and many other structures. And then also there is motility, there is the release of hormones and release of acid. Those all depend on the type of receptor that is present. So please don't go into detail here. Just know that these are all receptors. So you would expect the opening of the sphincter or the closing of the sphincter with the activation of the sympathetic system, whereas increased motility and relaxation of sphincters is all through parasympathetic effect. Okay, so constriction of sphincter here through one type of receptor, alpha 1, and then you would look at M3 as the one to bring about opposite effects. Decreased motility by alpha 2 and beta 2, increased motility by M3. Bronchioles, we have already discussed this briefly before. A beta 2 causes smooth muscle relaxation in the airways. That's how it dilates the airway bronchodilation. Okay, and the constriction of the airway is by the activation of the parasympathetic system using M3. This is an interesting situation here. This is only in the reproductive system of the male. You need the cooperation of both sympathetic and parasympathetic systems to bring about ejaculation and erection. So what I meant to say here is that typically what you see is a sympathetic action would be of one type, and the opposite effect is typically true of parasympathetic activation. So if you bring an increase in heart rate here, it's typically decrease in heart rate. But that's not how it works in the male reproductive system. Both sympathetic and parasympathetic systems are involved in accomplishing the erection and ejaculation. Ejaculation depends on the alpha 1 receptor. Again, this is one of those blood vessels that is supplied by alpha 1 receptors. Then erection is by the muscarinic receptor activation of the parasympathetic system causing erection. Then the ejaculation is through alpha 1 receptor and the activation of the sympathetic division. Now, bladder relaxation is using beta 2. Then that helps with the filling, constriction of the urethral sphincter, just like constriction of the digestive sphincters. Alpha 1 is involved in the constriction of the urethral sphincter. Contraction of the bladder wall uses M3. Now, sweat glands, as I mentioned, use the sympathetic supply. Increased sweating during sympathetic activation can be explained by the reason that it is supplied by the sympathetic division. And that's how you would accomplish this. And muscarinic receptor causing the dilation of the smooth muscle and increasing the amount of sweat being produced. Kidney increases renin secretion via beta 1 receptor adipocytes, lipolysis that's needed in the energy production during the activation of sympathetic system using beta 1 receptor. So this is a lot of detail to memorize. So whenever you're talking about one specific system, that's when you would apply these receptors and the autonomic structures that are associated with to understand the effects even more related to that specific topic. Now the summary of the activation of sympathetic system. This is the physiologic effect that one can easily notice. And we have talked about much more finer details before. This is just a broad effect that we are going to address here. Sympathetic activation often causes dilation of the pupil and increased alertness, increased breakdown of nutrients to derive ATP that's needed in muscle contraction, increased heart rate, increased rate of breathing, depth of breathing. So that is catabolism, increased muscular activity, cardiovascular, respiratory, and sweating. And one thing that is going to be inhibited during sympathetic activation would be digestive function. And another one would be the urinary function, decreased urine production, decreased digestive function is how you would see the effect of the activation of sympathetic system. Those would be ignored during fight or flight response. Blood supply is all switched to muscles, so minimal blood supply to the digestive and the urinary structures, and more blood supply to the muscles, the heart, the respiratory structures, etc. Now, the opposite is here during parasympathetic activation. Here, the drowsing type of scenario is what you want to think about. Pupil size is smaller than usual, and the opposite of what we saw with the sympathetic activation, that is decreased respiratory, decreased cardiovascular, and decreased muscular functions. Increased anabolic activity, that is the production of storage forms of energy like glycogen production, synthesis of lipids, proteins. So those are being typical during parasympathetic activation, and increased digestive and renal functions. Typically, think about it like this: food triggers parasympathetic activation. Food in the body would trigger digestive function, and that is how you have activated this parasympathetic system, which in turn activates the digestive function. Therefore, there is increased digestive function, there is increased absorption, there is increased storage of nutrients, and increased production of urine and release of urine are typical with parasympathetic activation. Now comes this important detail: who regulates autonomic functions? As you know, this is like vital function. Regulation of heart rate, blood pressure, breathing, all those are under the autonomic regulation. As you have learned, innervation itself is from the areas we talked about: cranial nerve 3, 4, 7, 10. So you talk about midbrain, pons, and medulla being part of the innervation. That is for the brain tissue. Now, spinal cord is where you talk about the thoracolumbar areas as well as the sacral part. Okay, T1 to L2 in the sympathetic division, S2 to S4 in parasympathetic division. So basically, you know that the preganglionic neurons originate from the brainstem area or from the spinal cord. Correct? So those are directly involved. Are there inputs that they receive from the higher centers of the central nervous system? Yes, limbic system has a serious input to the brainstem or the spinal cord, and therefore emotions can affect your eating or breathing or other functions that are associated with autonomic system. And hypothalamus is like the major structure that houses the autonomic regulation. That is the main one, and the signals from here are even regulated by the cerebral cortex. That is probably the higher center that brings the signals to the hypothalamus, which in turn communicates with the brainstem, with the spinal cord, and the limbic system contains hypothalamus. So hypothalamus is part of the limbic system. Therefore, they're all interconnected to be able to communicate with the preganglionic neurons affecting the autonomic function appropriately. So the regulation, although is directly from the brainstem and the spinal cord, higher centers like the limbic system, the hypothalamus, and the cerebral cortex have their inputs in the fine regulation of the autonomic